Yeah, I would like to invite Dr. Paranimuthu, who is a managing director of ARA Biotechnologies Pvt Ltd. He's an experienced managing director with a demonstrated history of working in the biotechnology industry, skilled with good laboratory practices, biosimilars, GLP, biotechnology, cell culture, and life sciences. He has a strong business development professional with a Doctor of Philosophy PhD, focused in biotechnology from Periyar University. I'd like to invite him on the stage.
Good morning, everyone. Before they open the presentation, maybe it is very important to thank the society and also our madam, our Agarwal ma'am, Sunita Rana Agarwal ma'am, and also the committee members Bhuvana ma'am, of course the many delegates, of course the the panels also. It's really very enlightening and very young enthusiastic audience I can see. So here actually I am telling you, I am going to construct 5-6 slides which I am going to talk about the company and the activity that we do. Then subsequently I jump on to the the areas. Of course gene therapy is something that very very interesting phenomena. I think you might have observed actually when drug controller was talking, he was mentioning a drug name and he told that actually per patient to treatment cost is 17 crores. Have you heard that? So that is one of the biggest success story globally now it is going on. So Indian rupee actually it is around 17 17 crore. So we'll go to the presentation straight away. So this is about our company. It's an IVD manufacturing company located in Chennai. So we have around 40 people working. We actually, you can go to the next slide. We actually involved in manufacturing all the IVDs both immuno and molecular diagnostic kits. You can go to the next slide, sir. Okay, yeah, it's there. So this is the main scientific team. Myself have about 20 years of experience. I have done a PhD as a CSIR fellow. I worked in both pharmaceutical and biotechnology. I was working in Bangalore also. Anthem Biosciences I was part, and also I was working for Tata. Prior to I was working for Dr. Reddy's Laboratories. Then with me Dr. Madhukanda, who is actually an IVD expert, who is in recombinant protein and molecular biology. Of course Dr. Vijayda, she is going to have one session. She is actually molecular biology expert and expert in developing assays, mostly on real-time PCR assays. So these are our quality compliances that we have, all the, as you was telling, the medical device manufacturing licenses that we have and all the quality compliances as per the ISO 13485. So some of the product that we manufacture for the human diagnostic is these are the listed product that we manufacture at Chennai. We also do manufacture the veterinary diagnostic kits also. That is one of the most upcoming area. These are the we both rapid as well as veterinary molecular diagnostic both we do. The panels which covers almost the livestock management and companion animal health and zoonotic pathogens and foot and mouth disease, canine parvo, rabies virus. All these tests that we have developed is all molecular diagnostic as well as the immunodiagnostic kits that we developed, both the ELISA, rapid and real-time PCR. So then we have one innovative device which has been developed for our country. Actually, that now that cervical cancer is it is more popularly almost more than 1 lakh women are affected. So we have developed a very simple self-sample collection and screening-based device that we developed, and which can cover almost all the 14 high-risk genotype models. That this is something that we are trying to work with many philanthropic organizations to screen as many as possible, the samples from our country. So with this, we have done a lot of sample. Now I come to the topic that which I am going to talk. So in the integrating genomics in routine clinical scenario, that as you all know that there and here there are some test kits have been that been used, and they used to tell that we are going to go for the exome screening, or next generation we are going to look at some Sanger sequencing mutation analysis and all. You might have heard some of the people they are doing. I'm going to take a very few case studies what are the challenges on each field, and also how it is actually implicating, what are the challenges, how that hurdles have been solved, and how it is being used in the regular routine practices actually. So when it comes to the, I'm going to kind of, see whether this genomics, whatever we talk, which can be practiced in the regular clinical practices. So this introduction, just for all our junior colleagues, wanted to give you, genomics is the study of, organism entire genetic makeup interacting to the environment, based on that we are actually studying the complete makeup of our gene, and which is the impact is definitely it is there on cardiology, oncology, and rare diseases, to find out they look at it, say that germline mutations. As you know that many of the mutations which are all coming under the introns, which is not affecting. Even now they are saying that intron mutations also can can cause the diseases. Then clinical care, definitely it is helpful in disease diagnosis and treatment, and also prevention. The importance is definitely it is guides as a personalized medicine, and it helps in giving a specific therapies, and also controlling it. So it is a very beginning of this genomic era. I would say the whole genome sequence project started in 2003. That time it was a very big process. Today, after 20 years, if you look at it, actually the system have changed, and the cost has been brought down, and lot many microorganisms, lot many genetic information, database, and data has been generated throughout the world, and everything has been compared. Now we are having enough amount of data. Yet actually I would say 25 to 30% only that knowledge of genomics that we have got into, which can be applied into the clinical scenario. So looking at these points, so the current landscape actually that subsequently the period of invention too, now that it is keep on evolving, many different platforms with the different technologies have come, and that the capacity of the machine, and the error-free sequencing patterns have been developed, which are all doing extremely good in the clinic. Now still there are some challenges also that we actually briefly look at it. So the current application, as I told you, cancer diagnostic, very much it is helpful in many of the tests, for the choice of treatment, they look at it, even a lung cancer, even a BRCA, breast cancer, they look at the treatment of options, whether they will respond to either for the kind of receptor tyrosine kinase inhibitors, or they will go for the other pathway based and all. Pharmacogenomics, of course, now the many drug companies, unlike earlier, there are some side effects are associated with clinical trial. So they go first, they study the genetic makeup of the particular volunteers whom they are going to be enrolled in the clinical study. Who is actually going to be enrolled? They will study, and they will segregate the pattern, the group of people who are susceptible to this, and they will give the treatment and clinical therapy. So the effectiveness will be studied very effectively. Mostly the warfarin, I think you all might have studied about the CYP2D6 enzyme. Even today it is available. Before taking this drug, actually they will ask us to check that mutation is available. That kind of a pattern is there. So this drug can retain in the body for a very long time. This is one of the tests. Even today, many of them, they are taking it before they are going for it. And of course, for the parental screening that you all very well know. Even nowadays, not only this downstream and trisomy, even many people you would know, our relatives and all, they go for the IVF, IVF when they go. Even that time, actually, before they actually, they go for it, they can completely do the post the ovulation and even pre-ovulation. Both they can study the complete genetic makeup, so that most of the inborn error of metabolism, child based genetic diseases, that can be avoided actually before even they conceive actually. That is also the technology has come. So this is briefly I touch upon. So there is a 45 years patient who are having the BRCA positive, but they are having the metastatic breast cancer, but actually it is an ovarian cancer. It is metastasized to the breast cancer. Identified actually there is a positive, then they have been going for the treatment. It is giving better results. Even other sites, when you study genetically, you are able to treat it. That is one. And success story, population wise, actually, if you look at it in the Israeli population, there is one of the population which having a very severe impact on this BRCA1 gene. They are very susceptible for the breast cancer. Now most of the population actually, as you can see, Israeli, Ashkenazi, Swiss population, they are regularly studying that population, and before even they go into the adolescent stage, the study, they actually do the treatment and prevention. Same like the human papilloma virus, how we are trying to study before even that the viruses get into the cells and transform the normal cells into cancerous cells. Then the challenges what all there in the oncology, if you are looking at it, as I am telling that oncology is a very excellent area to look at it and study and develop the technology and develop the drug model that we can test it as a personalized therapy. However, there are some challenges cost accessibility is there, and of course it is varying country to country. Then result interpretation, again too much of data is generated, still that correlatively because of cancer actually it is highly heterogeneous. There are one tumor is there that can have a 3 or 4 even more than that type of different genetic makeups, and it's a sequential mutation, it's keep on happening as soon as it becoming cancerous. So based on that it can actually do. And of course the healthcare infrastructure, hospitals are not really focusing on to bioinformatics and data development, those things. If we do that, then these challenges can be managed. And of course pharmacokinetic, again this I have already told you. CYP2C9 also like a palmosome which is undergoing for the salient features, which is the case study where the mutation alters the fewer complications to it. Then the challenges which have variabilities in genetic influence, and the clinician knowledge gap is also a challenge in pharmacogenomics, and insurance barrier. When cost is coming, other country actually these genetic tests are actually covered under the insurance, so the cost is not a problem. But here that is one of the challenge where in India it is still not under covered. And of course the rare disease it is in the inborn error of metabolism where you can see the very clearly and save the patient before they even look at it. So the 40% they can actually clinically studied and they can be recovered before that clinical outcome comes, because the Rett syndrome actually it is something after 6 to 7 months later it appears. But the early stage itself in inborn we study, we can control it and we can take it forward. These are all some of the clinically relevant. And the same thing this is here also, that this is what the drug I was talking about actually. This is a drug which is launched and it is developed by a European company, Novartis. This is actually 17 crore actually required for doing a treatment with this drug for the spinal muscular dystrophy. This is one of the first gene therapy actually. Since our our madam also, Sunita ma'am also, she is working on a very fantastic technology which is similar to that. When we interacted to her, we were really the global technologies are actually the conceptualization ideas and the developments are actually being done by our our Agarwal hospital. So can we move on? It's not moving. Can you move to next slide? Yeah. So the rare diseases, what is the challenges? Again, cost affordability and very early stage actually the patient the babies has to be brought to the genetic counseling and the treatment, and the data sharing challenges will be there. Of course, the many identified the variations are remaining still, because many of the rare diseases are, we don't have so much population. Now if we say in a cancer patient you wanted to study certain things, for example non-small cell lung carcinoma, if you wanted to study, you can find a very large population. For example, the rat disease which I was talking about, in whole India per year actually you will get around 4,000 to 5,000 patients only. So your sampling going to be very rare. So the variability, the study has to be really developed it in a more systematic way. Yeah, next slide. So cardiovascular risk also same way only, the familial cardiovascular risk will be there, but they may not know. If we study that, then food regulation and they can monitor it very well. So very easily they can be saved from the atherosclerosis and other cardiovascular events. You can go to next slide. So that again success stories, there are many countries, they have done a very huge amount of data they have generated, and the 25% of the rare disease have been studied and it is accessible now. And of course the Iceland has again further they have studied more on the rare diseases. So there are some ethical issues with respect to sharing the data, handling the patient sample, and the handling the data and interpretation, and also the treatment advices, but which need to be addressed actually. Yeah, you can go to next slide. So the barriers, these are the barriers, the infrastructure and cost, and the example developing countries which are as kind of country Of course, we are we become clinically very much we are having a lot of the infrastructure in the private organizations which need to be integrated with the clinics to do practice on day to day, regular clinical practice. And the implementing, again, standardized guidelines required, and the trainings has to be then, doctors has to be integrated. Most of the time, the biotechnologists and biologists will be doing it one side, all the experiment, and which will never be translated in the clinic unless until the doctors have been integrated. And the case studies rural health clinics and who are trying to do this, lack of resources. These are all some of the areas why actually we are not able to bring the genomics into primary care. Yeah, you can go to the next one. So, the future that can be emerging technologies are CRISPR-Cas, gene editing, RNA based therapies could be a revolution for the even for the therapy as well as for the diagnosis also. Then sickle cell therapies, as we was talking about, sickle cell therapies are also already they have been addressed using a genetically sickle cell, thalassemia, all of them are being genetically approached now. Then population wise screening is required to generate more data. And definitely AI has to be integrated because that has very powerful, and it can even look at it in a much more holistic sense. It can look at it unbiased manner. Actually AI can address the issues, and data interpretation shall be much better. So the AI is playing very important role, and the machine learning and AI algorithm, and these are all bringing a very excellent system where actually the doctors as I was recently I have been hearing regarding the HPV we are working. There is one Hyderabad company. We are actually working at the virus. Actually we are looking at only the virus, whether it is infected or not. But they are going to the next level. They are looking at even the cell transformation. Either it is in a cervical neoplasia stage 1, stage 2, stage 3, which is actually integrated with the AI. Actually they are studying the cell morphology and giving the result. So when you combine both virus and the cell transformation, this will become a complete solution for the HPV screening. This kind of integration is very important now. So the keynotes, the takeaways are genomic already making profound impact in the biology past 20-25 years. After this NGS pattern, it is really tremendously grown, and many platforms have come. All that accuracy data has been generated by various companies which have developed the platforms, and that we need to have the cost and data interpretation knowledge has to increase. Ethical practice is required. Of course, the clinicians has to be integrated to this process to practice it regularly. So we have to more read and integrate and innovate it. So thank you very much, one and all. If you have any questions particularly, maybe I can take. I hope I timed up. Yes. Any questions? Thank you, sir. Time okay. Very good.
Muthu, I have a question on behalf of the whole students community. You see, most of our audience here are students, and I am seeing you from the day when you were like them. And one thing I have to appreciate about Muthu is a student time entrepreneur. So his entrepreneurship started when he was like you. Now the question is, when he has become a world-renowned entrepreneur, it is his time to give it back to the students community, and that's what he is doing now. My question to you, Muthu, through RO Biotech, what is something the students can benefit by utilizing the innovations which your company is doing here, which can help them to make their projects successful?
Absolutely. Yeah, thank you very much. Actually, as he rightly has pointed out, at many institutions and universities, I spoke about the bio-entrepreneurship and the development and other things and all. This is something to very strictly adhere to this International Society of Gene and Cell Therapy. So I have given this presentation. But truly speaking, as he was telling, from the days when in my college days, I was very clear what we have to do. But how you can actually utilize Aura Biotech is, there are many innovative projects that we have been working on a social cause. For example, sickle cell anemia We are developing a very, very simple paper-based kind of detection system. Similarly, we are having an isothermal PCR technology, which we are collaborating with one Korean company for developing a technology for tuberculosis detection. So there are many the unmet medical need and challenges which are there prevailing in India. We are working. Since we understood that actually the women in India who are really suffering, and almost 1.3 lakh women per year we are losing because of the cervical cancer. So we felt that this is something beyond our commercial activities, beyond our business. This is something that the social cause we must address this actually, because most of the test kits which are coming from the abroad, we import the the HPV screening test are the way high actually. Now normal women's, like for example COVID also, the earlier COVID time we started with a very big price 600, 800 and all, then finally it come to 100, 200. Similar way, can we translate this HPV screening? This was one of the screening. You are all most welcome to volunteer. We were actually planning to do a mega screening of maximum possible states. We are going to screen the women for the early stage. If you are detecting the HPV itself, early stage, 30 to 40% of women can be saved actually. So in this mission, you all can participate and you can write to us. So in any of the projects, anything, guidance, suggestions you required, how you can build a startup organization, that we can definitely help you, provided your idea should be very good actually, which can be immediately made it as a company, it can be a profitable company. Otherwise, my advice would be, kindly please go to an industry and work there and learn about all the departments. It is not only the company running, the entrepreneurship, it is not only about the innovation and technology. This has so many integral departments which has to be included, including finance, sales, marketing, manufacturing, regulatory, and each and everything you can learn if you go and work in a very big professional organization. That was my plan. When I was studying college itself, I wanted to be an entrepreneur, but I have not done that immediately. I went to Dr. Reddy's laboratories. I could 7 years I worked there. I went to Tata. I worked there. Then I went to Biocon. I worked there. So this is how I learned the experience. So we are able to sustain. So this is one of the idea I would like to tell the students, that go to your professional companies and then you start your idea. So you will never have challenges actually. You can continually, you can grow. Yeah.
Thanks, Dr. Paranimuthu.
How do you see the integration of genomics transforming routine clinical decision making? And what role does patient data privacy play in ensuring the success of the transformation?
So the patient privacy and also the translation, which is I have already spoken in the presentation, which is one of the challenging thing. But actually there are some ethical policies have already been given by the government, where what area need to be declared, openly declared, which cannot be declared. Like that, there are 2 classifications have come already. So which is now everyone is able to take, but still the data sharing, still it is a confidential. Yeah.
Okay. Thank you so much, Dr. Palanimuthu.